Now that you kind of understand DNA replication, with DNA replication, it's all about base pairing. That's it. All about base pairing each side, and then once you base pair each side completely, you add a strand to that original side. We call it semi-conservative replication because the parent strand is conserved, isn't it? It's bred, and then a new strand is formed to attach to that parent strand. So on each side, a parent strand is conserved, it's bred, and then a new strand is attached to it. So we conserve the parent strands. Once that we have kind of gone through DNA replication, we've doubled the number of chromosomes. Now we get ready for all the machinery that will move the chromosomes to the proper location. This happens in the second gap phase as we get ready for cell division. Because remember, we somehow need to grab these chromosomes and then pull them apart. Because we don't want one cell to have 2 copies of chromosome number 1 from mom and dad, right? We want it to be split apart. Now, in cell division, it starts off with prophase, then it goes to metaphase, anaphase, then telophase, and it finishes off with cytokinesis. There's that mnemonic, right? IPMETC, right? So here is, right, I for the whole interphase area. Interphase is this whole system right here. Starting with G1, it includes S, and then with G2. That whole region is called interphase. So I for interphase, P, prophase, M, metaphase, A, obviously, anaphase, T, telophase, and then C, cytokinesis. That is a mnemonic that a lot of people use to memorize the sequence. Now, what you need to also remember is what happens in that sequence. So let's take a look. We've already doubled the DNA, and now we start seeing this prophase. Prophase right here, let me see if I can make it a little bit bigger so we can see it. Okay, too big, too small. So in prophase right here, that's when we start seeing all of the chromosomes, right? So we start seeing it slightly in prophase, and whenever we see chromosomes with 2 arms, like we see here, right away, that chromosome has undergone DNA replication. So what we're gonna do is this, double the chromosomes and then align them a certain way. So we're gonna kind of go through this, and then I'm gonna show it to you on some videos, some animations, and also on the board right here. So what do we see? Well, when we're looking in a cell that is not ready to divide, so the cell is just undergone DNA replication, it's not ready yet to divide, we don't really see the chromosomes yet. You only see the chromosomes and the DNA in prophase. Once everything is set and you're ready to divide the cell, that's when we start seeing the chromosomes. So early prophase, we start seeing the chromosomes, and then you also start seeing the machinery that will kind of guide the chromosomes into place. Because what we want to do is put all the chromosomes right in the middle of our big cell. As it's right in the middle of our big cell, we can just pull them apart, separate them. And then one side gets one chromosome, the other side gets the other chromosome. So each side will eventually become different cells. And that's what we see here. In the early prophase, these spindle fibers and the centrioles move to opposite sides, 180 degrees apart, right? So they move. They're originally, right, these centrioles are gonna be right next to the chromosome. Now they're gonna divide, and then they're gonna split at 180 degrees apart. At 180, you are physically as far apart as possible from each other. As they move apart right here, they start sprouting these fibers. These fibers then connect, and they connect specifically to the middle of each of the attached chromosomes. Right? The middle is called the centromere. So you have these fibers that kind of sprout up, they attach to the central region, and the reason why the attachment is important is the attachment allows you to line up the chromosomes. And that's what we're gonna see in the second part right here. Again, prophase, you see these fibers, they start attaching, they start moving the chromosomes up towards the middle. As you move the chromosomes towards the middle, the chromosomes all lay one on top of each other. That's gonna be very important, all right? I'm gonna write it out a little bit better so we can see why it is important. So here, notice that the chromosomes here, the way they're lining up is incredibly important. In metaphase, all the chromosomes are in the middle of the cell. So metaphase, middle, or medial. All the chromosomes, 1 to 46, right? Now these 46 chromosomes each has double arms. All of them are right down the middle of the cell. And what happens then, right, in anaphase, we pull the chromosomes apart. Remember, when you have two arms right here, one arm was original, the other arm is a copy, right? This one right here will be from dad, this one right here will be from mom, chromosome 1. So mom's chromosome 1 right here, you'll have two copies. Dad's chromosome 1, you have two arms, you have two copies. That's true for all 46 chromosomes. They're all lined up there, they all have two copies. What happens then? In anaphase, we pull the chromosomes apart. As we pull it apart, one chromosome number 1 from mom goes to this side. The other chromosome 1, that is a copy, goes to the other side. Each side will turn into a different cell. Chromosome number 1 from dad, right? One arm goes to one side, the other arm to the other side. What happens? Well, then each cell right here, this cell, will get one copy of chromosome 1 from mom, one copy of chromosome 1 from dad. Again, exactly what we want, right? One copy of one chromosome from one parent. So mom gives you 23 chromosomes. Dad will give you the other 23. You shouldn't have the same two copies from one parent, right? You shouldn't have two copies only from mom. Or two copies of chromosome 1 only from dad. If that happens, that's a mutation. That's not good. So by lining it up, all the chromosome 1s, all the chromosome 1s, all the chromosome 2s, all the chromosome 2s, lining them up that certain way in the middle, when we split it apart, this copy of chromosome 1 from dad goes to this side. This copy of chromosome 1 from dad, other copy, goes to the other side. Chromosome 1 from mom, chromosome 1 from mom. What happens? Right, this cell right here will get one copy of 23 chromosomes from mom and the other copy from dad. Same thing with this cell, right? Exactly what we want. One copy of each 23 chromosome from each parent, not from the same parent. Now, that happens in anaphase where we kind of pull everything apart. In telophase, everything is already pulled to the proper axis or proper pole. So now we're going to start dividing the cell in half. So when anaphase occurs and we bring the chromosomes in the right direction, in the right place, now we're going to restart forming a new nucleus from it. And we're going to start to cleave the cell in half. That's the thing with the telophase. We start that cleaving, we start forming the nucleus in telophase, right? So new nucleuses form on each side. We're going to start pinching the actual cell in half. In cytokinesis, right here, we have completely separated. And now the cells are completely separated from each other. These two cells should be identical to each other. Now, we are perfect. All of the chromosomes here and all the bases on the chromosomes will be identical, right, to this other cell. These should be just a replicated cell. So everything should be identical when I compare this cell to that one. Now, if I'm completely perfect and the cells undergo this process perfectly, these two cells that we just formed will be identical to the parent cell as well. That would mean there's no mutations, the telomeres aren't shortened, and now This original parent cell will be identical to the 2 daughter cells that are produced at the end. That would be if everything works out perfectly, and we hope it does. Usually it does. I'm going to show you an animation, and as I show you the animation, keep in mind that it is online